What's good, everybody? Welcome to a video on the mechanism of Ozempic. Now, Ozempic is part of a larger drug class called GLP-1 agonists, which are based on the naturally occurring peptide glucagon-like peptide 1. This is a drug that primarily in the past has been used to treat type 2 diabetes by improving insulin sensitivity. However, these days, mostly people know it by its ability to help people lose weight, by decreasing appetite through gastroparesis and inhibiting their desire to eat. The most common side effects that nearly everybody will have to some degree is GI disturbance, maybe not quite to the level of nausea or vomiting, but those are certainly among the most common side effects. You also can have hypoglycemia, meaning low blood sugar, and then pancreatitis and an increased chance of pancreatic and medullary thyroid cancer. After we go through the mechanism, it'll be pretty clear why you might have an increase in pancreatitis or pancreatic cancer, primarily because you're just making your pancreas work a little harder than it normally would. And this is not medical advice. This is not the point of this video is not to encourage people to use Ozempic, but my advice is if you do use it, titrate up over weeks to diminish the probability that you have side effects. And remember that this drug doesn't work unless you work. So if you're not actively trying to change your exercise regimen and your lifestyle, there's really no point in going on this, as those people are most likely to regain all of the weight that they lose. So the most effective way to help keep your results are to change how you eat higher protein, and start weight training. Help hold on to any muscle that you may lose as you're losing weight, because if you lose weight too quickly, you'll end up cutting into muscle as opposed to fat. Now, getting into the background of how GLP-1 agonists were created, essentially they took the natural GLP-1 compound and they made 3 different changes to the molecule at the 8th, 26th, and 34th amino acid. So you can see all of these little balls are just different amino acids that make up the larger protein that is GLP-1. And overall, these changes were made in order to increase the half-life of the compound. As you can see here, normally GLP-1 only lasts 1 to 2 minutes in your body to exert its effect. But because of these changes that were made, Ozempic or semaglutide lasts up to 1 week in your body. And so the 2 things that were done, you have an amino acid substitution here, which protects against DPP-4 degradation. So this DPP-4 enzyme is what normally degrades GLP-1. In this case, Ozempic is immune to that. And then these other 2 changes both help to allow Ozempic to be bound by albumin, which is a carrier protein in your blood. Now, before we get into the very specific parts about the mechanism of Ozempic, we have to understand the process of insulin secretion in the body. And so I have 2 different images here. They both show the same thing. There's just certain things that I believe highlight the idea that we're trying to convey more effectively. I mentioned salty calcium banana at the top because you can think about insulin secretion in a similar manner to action potentials in neurons. I do have a previous video on how a neuron works that would be helpful in thinking about this. But the neurons case, we think about them as salty bananas because they have a lot of potassium on the inside of the cell and a lot of sodium on the outside of the cell. In this case, when we're talking about pancreatic beta cells, you also have potassium on the inside, but instead of the salt, we'll think about you have calcium on the outside. And so what happens, we'll start on this left side. You eat food which has glucose in it. Glucose is just sugar. So sugar is transported via the GLUT2 transport into the body and goes through glycolysis and the Krebs cycle and the electron transport chain to produce ATP. And then an increase in ATP will inhibit ATP-sensitive potassium channels, which will cause less potassium to move out of the cell, which means that the negative 70 millivolts that the cell is normally at will increase drastically. So kind of an action potential will occur and the cell will be depolarized. That increase in voltage causes this voltage-dependent calcium channel to open. Calcium flows in, and this calcium causes a release of these granules containing insulin, and these granules come and they bind to the membrane of the cell, and they release the insulin to go all over your body and essentially act to tell your cells to release a bunch of these glucose transporters to uptake glucose into the cell to make more energy. So I know that was a lot, but I think between these 2 diagrams there should be a pretty good understanding of what's going on. And now, when you take either Ozempic or the GLP-1 hormone acts in your body, it helps to increase the sensitivity and the likelihood of you releasing insulin. And so instead of having, say, a negative 70 millivolt resting membrane potential, this will increase it to maybe negative 50 or negative 40, so that there's a lower threshold of glucose that needs to come in to depolarize the cell. And furthermore, once it is depolarized, it's a more intense depolarization, meaning there's more calcium that comes in, which can release more vesicles containing insulin. Furthermore, it also inhibits the potassium channels that help to bring everything back to an equilibrium. And so you can see here, we'll follow along in the diagram, the GLP-1 will bind to the receptor, increase adenylyl cyclase, which increases the conversion of ATP to cyclic AMP, and cyclic AMP will activate protein kinase A, which will inhibit this potassium channel, meaning you keep more potassium in the cell. Because the potassium is positive, that will increase the resting membrane potential, and it also activates potassium coming into the cell, so the resting membrane potential will become even more positive. So once again, whenever glucose does come into the cell, create more ATP, and there's a depolarization, there will be more calcium released, more insulin released. Furthermore, on the other hand, cyclic AMP also activates this EPA-C2, which helps to release even more calcium from the endoplasmic reticulum. So in a variety of ways, the GLP-1 agonists are able to increase the voltage, resting membrane potential, and also increase the duration of the action potential. And then last, which is not actually on this slide, it does inhibit glucagon, which is the hormone that increases glucose when you have low blood sugar in your body. Unclear exactly if the GLP-1 agonist directly inhibits glucagon release, or this is just done because insulin itself, when it's released, inhibits glucagon release, because they're kind of opposite hormones. Additionally, GLP-1 agonists inhibit the hunger mechanism in your brain. So once again, we have a diagram here on the right, which we'll kind of walk through. Essentially, you take in the compound. Normally, GLP-1 is released in your intestines when you eat food, and it will bind to the GLP-1 receptors on the vagus nerve, and the vagus nerves will convey a signal up to these different regions of the brain called the AP or the NTS, which will ultimately transmit inhibitory signals to other regions of the brain. And these inhibiting signals are GABAergic transmissions, meaning that it uses the neurotransmitter GABA to decrease the resting membrane potential, making it less likely to fire. So it's inhibiting the activity in those brain regions. Those particular brain regions you can see here, the ARC, PVN, and SVO regions. Those stand for the arcuate region of the hypothalamus, the paraventricular nucleus of the hypothalamus, and the subfornical organ of the forebrain. And these are all regions that are known as either orexigenic or anorexigenic, which you can see down here, which orexigenic just means that it promotes an appetite. So if it's anorexigenic, it means that it decreases your appetite. If it's orexigenic, it increases your appetite. Furthermore, inhibition of any regions that release high amounts of neuropeptide Y and agouti-related peptide are also inhibited by the GLP-1 directly and through this vagus nerve signaling pathway. And both of these are just hormones and peptides that have positive orexigenic effect. Neuropeptide Y in particular is often cited as the peptide that encourages carbohydrate cravings. Last, the POMC and CART neurons, which are also in similar regions of these, are activated by GLP-1 agonists, and these actually are anorexigenic regions, meaning they decrease appetite. It's a little bit esoteric what happens moving on from there once you get this deep into the brain and what pathways mean what and what signaling means what in the regions of the brain, but I thought this was sufficient for this video. Thanks for watching. I hope you learned something today.